Hello, my dear little sister. Since you're curious about what I was doing for my practical class, I will be explaining to you about DNA microarray and how does DNA microarray works today. So, what is DNA microarray? A DNA microarray is a collection of microscopic DNA spots attached to a solid surface. So, you can see there's a chip which is fluorescently labeled with red, green, and yellow. So these colors indicate the expression of large number of genes. So this expression helps to indicate which type of genes are upregulated and downregulated in a particular cell of an organism, of a living organism, I would say. So you must be wondering, what does it mean by gene expression? Basically, what does gene expression means is that which gene is upregulated or downregulated, or in a simpler term, you can say on and off in that particular cell. You can ask me, why does it have to have this kind of gene expression in a human body or in an organism? Okay, this is basically because we don't want unnecessary products to be produced. For an example, due to certain gene upregulated and certain genes that is downregulated, our eyebrow does not grow longer than a certain limit compared to our hair growth. So just imagine if we do not have a gene to control our growth of eyebrow. So this is why it is vital to have this kind of gene mechanism. So moving on to DNA microarray, you might be wondering what genes are upregulated and downregulated in a cancer patient versus a normal human being. This can be identified by DNA microarray experiment. So this is an example of the experiment that has been conducted to identify what are the genes that are present in a normal cell and a cancer cell. So researchers will first remove the normal cell and cancer cell from the human body, from the infected and uninfected human body. So, and then the mRNA is extracted from each sample. The mRNA is then reverse transcribed into complementary DNA, which is also known as cDNA, by reverse transcriptase, which is an enzyme. And cDNA is more stable than mRNA. The infected sample is fluorescently labeled red, while the uninfected sample is labeled green. At this point, in many microarray experiments, the two cDNA samples are combined. The combined sample of fluorescently labeled cDNA is applied to the microarray chip. The cDNA from the sample binds to the complementary sequence of DNA bases on the chip. This chip is then inserted into a laser scanner to electronically capture the data. The chip is scanned with laser, which activates the fluorescent dye in the cDNA sample. This information is captured by a computer. It calculates the ratio of red and green on each spot, which indicates the genes that are expressed in the normal cell and cancer cell of a human being. So, in this diagram, you can see a chip which consists of red, yellow, and green dye, which is fluorescently labeled yellow, green, and red dyes. So, the yellow indicates that the genes are present in both cells, which is both normal and cancer cell, whereas the green indicates the genes that are only upregulated in normal cells, and the red color indicates the genes that are only present in cancer cell. So from this experiment, you will be able to distinguish between the type of genes that are upregulated or present in a cancer cell versus a normal cell. So from this you would know which genes causes cancer and which does not cause cancer. And you would know which genes are present in a cancer cell basically. So I really hope that I did justify to your question by explaining to you what does DNA microarray means and how does DNA microarray works. If there's any question that you would like to ask me, you can ask me. Okay, my dear little sister, thank you so much.